I'd like to thank the organizers for inviting me to this exciting event, and I've learned a lot. So my group works in the cancer systems biology. We generate a lot of the data. We are not physicists, not the mathematicians. So hopefully in this talk, I'll be showing you some of the data-driven observations that would lead to the indecisions in our group, but hopefully that would lead to the questions and interactions. The key things that I would be covering in this talk are that what regulates the transcriptional state dynamics, what alters this transcriptional state dynamics, and ultimately, if we look into the tissue context, what influences this orchestrated state dynamics across the cell types in that tissue niche. The system that we are working on and the example that I will be showing is the urothelial carcinoma. It is the most expensive cancer to treat. The quality of life is really bad after the surgeries. And also it is an exciting and interesting system to study because we can learn a lot about the disease biology in a non-invasive way, and there is still very, very limited therapeutic opportunities. I want to thank first the lab, who are the real engines for the innovations. I come and give the talks, but they're the ones who really make the things happen in the lab. So the talk that I would be showing or the work that I would be showing are essentially of a very smart graduate student, Luisa, and a staff scientist, Antara, in the lab. So this work started with a very simple overarching experiment and the analysis to see that if we examine maybe the bulk tumor genome sequencing or the transcriptional sequencing data, what drives the intratumor or the intertumor heterogeneity in the gene expression space. And then at the time, the single cell sequencing data also came up, and we went on to examine that what drives is intratumor and intertumor heterogeneity and what are the sources of the variations. And once we overlaid the cancer hallmark associated signatures, it appeared that EMT appears to be a very strong factor that is associated with these intrapatient heterogeneities. There are cells that show more E-like signatures. There are cells that show more M-like signatures. So then since in the single cell data, we can reconstruct the evolutionary histories to some extent across the clones and the subclones, we resolve the single cells entities into the major clones and subclones as far as we can go, and then ask that whether the sister cells are more likely to be more E-like or M-like, or whether there is a switching that is happening across the space. But this is an observational data from a single snapshot of the tumors. We cannot really recapitulate what might be happening in the real time. So this data, again, is interesting observations, but we really need to examine into a system. So for that, in the lab, we went on to, also one more thing, once we look into the phenotypes of the cells, we realize that it is not just EMT, but these signatures are also associated with maybe stemness and associated with other hallmarks, so that we can actually aggregate these phenotypes and think that these are reflective of the phenotypic states of the cells. And so instead of looking at the cancer hallmarks specifically, if we see them as a form of the states, it would be easier to explain. And so we went on to see that whether we can get a laboratory system that can, to some extent, recapitulate what we are observing in the human tumor so that we can alter it in a way that we want. So here we looked into two specific cell lines. One is called T24 that spontaneously in the laboratory conditions make two different type of the phenotypes. One is called holoclones and one is called miroclones. The holoclones are more stem-like, they are more mesenchymal-like, they can continue to grow for a longer time. And the M-like cells, or these miroclone-like cells, usually are more differentiated phenotypes and they grow less, but they can switch from one to the other in the laboratory conditions. So what we did is that we did the long-term cell culture, and also we did the live cell tracking, and at the end of the experiment, we also did the single cell sequencing of these cells. And so here is what we see from the live cell imaging data. The cellular phenotypes very clearly show the holoclone and the miroclone-like phenotypes as we would expect. And as we grew the cells over the time, we could see that those colonies of the holoclone started to have more M-like cells. And then over time, after several days, they were more in the equilibrium. So we went on to see how they approach the steady states and what would be the cell transition dynamics that could be there. And that could be an interesting observation in itself. But in the sake of the overall storytelling, let me move from here to see what we see from the single cell data. So once we did the single cell sequencing of the same cells, we could split, or we could overlay the gene expression signatures on top of them and see that the cells also recapitulate at the transcriptional level the E-like and the M-like phenotypes. And once we look into not just the overall signatures, but also specific gene markers like CDH2 or the CD24 that are the more traditional markers of the epithelial and the mesenchymal phenotypes, the cells form into these two different axes. And so these give us the idea that this is an interesting experimental system that perhaps may be able to recapitulate what we are observing in the tumors and allow us to find out what might be regulating these cell state dynamics in the in vitro system. So then that gave us to the obvious question that what might be the transcriptional regulatory network, if there anything that exists that would be driving the cell state alterations in this setting. So we worked with Mohit Jolly from ISC, and this was a very, very rewarding collaborations. Essentially, we started with all transcription factors in the genome, and then we asked which ones are expressed in the urothelial cell types, which ones are associated with the EMTs, and in our this large-scale experimental data, which ones are showing the high level of variance, and accordingly, we started to have a smaller transcriptional network, and then we started to remove one transcription factor at a time until we got the minimally sufficient regulatory network that we think can recapitulate the alterations we are seeing. And that includes 3 genes, SMAD3, KLF4, and PPAR gamma. And as you can see here, that these show very segregated expressions across the cell types. And if we just move to the last slide, we can see that that actually overlays very well with the EMT phenotypes that we observed earlier. So then we then went on to ask that if we suppress the expressions of these genes using siRNAs, does that alter the cell states in a way that we would expect? The answer is yes. And also, when we looked at the RNA velocities, we could see that it is not only altering the cell states, but the intention of the cells to move to the next state is also pretty evident there. So in summary, from these slides, we can think that these 3 gene regulatory network is recapitulating the transcriptional regulatory makeup of the cell states in a way that is kind of sufficient for us to explain what we are observing. But, and also we did the spatial transcriptomics on a small set of samples at the time, and we could see that the EMT phenotypes are being associated with the expressions of these genes, which suggests that this is not just a laboratory observation, this can be translated into the urothelial carcinomas in vivo. So for the sake of the interactions, I thought that like the few questions that keep us up is that, are these 3 genes necessary and sufficient, or is there a larger network of the transcription factors that we need to take into account, and what might be a sufficient numbers or not? And also, if we look from the urothelial carcinoma to other cancer types or other organ types, are there other set of the transcription factors? Of course, in the literature, we have seen many, many other transcription factors being reported for other cell types. But are these reflecting the similar kind of the domain of the transcription factors, or the similar universe of the transcription factors that are driving these phenotypes? And just these 3 genes are probably the manifestation of that larger regulatory modules that one would be thinking of. And third, to what extent this transcriptional state dynamics is getting buffered, and would that have an implication for the adaptation and for the robustness? Of course, for the cancer cells, we do not want it to have enough adaptation and robustness. We want to have the opposite. We want to alter it. So that led us to think that how we can alter these transcriptional state dynamics in this system. So we did the radiation of the cells. It is easy to do, and it's also relevant for the urothelial carcinomas. And we could expect, and as we have expected, the epithelial and the M-like cells do respond to the radiation in the different ways. They have the different sensitivities. But also, interestingly, upon the radiation, the cell state dynamics alters. There are more M-like cells which are more radioresistant than the E-like cells. So we think that this might be a system for the adaptive resistance, even when there is no acquired mutations that would be showing up. And so this can be itself a long follow-up and the engagements, but for the sake of the interactions, I thought that this can lead us to think about whether with the radiation we should be thinking about maybe the combination therapies, and the second target is not necessarily what is going against the actionable oncogenic roadmaps, but essentially something that is altering the cell state dynamics. But while we were thinking about that, we went on to think that what the nature can inform us. Can we learn anything about the cellular phenotypes by looking into the tumor microenvironment? What might be the nature's way of altering these cell states? So for that, again, we did the serum starvation in the lab, and we could find that the serum starvation alters the cell state dynamics, and there are more M-like cells and the E-like cells that start to show up. And when we did the spatial transcriptomics, we could see that these EMT phenotypes are associated with hypoxia. We did know that the tumor cores are more hypoxic and the cells are much less replicative. They are much more quiescent, but it is interesting to see that these observations we can reflect upon from this setting. But that gives us to think that whether when we think about translating the laboratory-based observations to the in vivo settings, whether there are any kind of transcriptional or the functional buffering that might be happening that we have to think about, and to what extent the tumor microenvironment might be driving that effect. And that's might be one of the reasons why the therapies fail so often when they are so successful in the cell lines and in the labs. So that made us to move to the last part of my talk, that how do we learn about the cell state dynamics and the microenvironmental dynamics in vivo, and what might be the drivers that would influence the choreographed the cell state alterations across the cell types at the same time. So for that, we started with the same H&E slide that our histopathologists very nicely annotated across different regions. We overlaid these EMT signatures, which are cancer-specific, and we could see a heterogeneity across the slide, which would show that some parts are more E-like, some parts are more M-like. Of course, some regions are also the non-tumor cells that would be there. And when we overlaid the FGF signaling, which essentially comes from the fibroblast, we could see that that also shows a spatial heterogeneity in the sample. And if you see, so the small arrows there indicate where the ligand enrichment is and where the receptor enrichment is. So you can see that that upper left corner is where we see a lot of the FGFR signaling, and maybe that's where we are seeing a lot of the EMTs. This is an anecdotal example, but it shows that there might be something that could be happening there. So that would link us to think that whether these alterations are related to each other, whether we should be thinking about the tumor biogeography more seriously than what we are doing now. And that led us to think that, okay, first, one sample is not enough. We have to do more samples. But as a small lab, I could do only a small set of samples from the different subtypes of the urothelial carcinoma. And also, we came up with algorithms. We came up with an algorithm that can analyze the spatial transcriptomics data to identify the tumor-rich and the stromal-rich regions and find out the tumor-stroma local interfaces, and then go on to ask that what might be the cell types that might be around those tumor-stroma local interfaces, both in the tumor-rich region, in the stromal-rich regions, and if we classify these interface regions, would we be seeing some regions as being more active and some other regions that might be less active in terms of the functional activities? And when I say activities, we started to look at the transcriptional functionalities, but when we overlaid the cancer hallmarks, we could see that the active regions automatically show higher activities of the EMTs, the cell migrations, and so on. So that gave us a feeling that maybe the active regions are really what we are thinking about, the invasive regions, and the less active regions are the ones that are much more quiescent. And then we went on to ask in the module E and F that what might be the signaling that would go on across those interfaces, and can we learn something from there? So here, just the snapshot of those tumor-stroma local interfaces. The left side ones are the ones that are the urothelial carcinoma. The right side ones are more of the neuroendocrine, the ectodermal subtypes, and the keratinoid subtypes. And you can very clearly see that the amount of the marbling we see, or the amount of tumor stroma intermixing we are seeing, is very different in the regular urothelial carcinoma and the other subtypes. So that means that the tumor stroma architectures are very different. When we overlay the activity scores, we could subclassify these interface regions in terms of active, intermediate, and lowly active regions. And we could see that those are not being randomly distributed throughout the interfaces. Some regions show the concerted level of activities and other regions show the less concerted level of activities, even though we did not impose any of the spatial information while we tried to classify these regions, which suggests that probably we are identifying the real systematic active regions and real systematic inactive regions in the tumor stroma interfaces. And when we look across these samples, we could see that we do see a lot of intra and intertumor heterogeneity. For instance, the third sample, S3, has a lot of active regions. Maybe a sample S9 has, or S8 has got some active regions, some inactive regions, and these are spatially segregated. When we look into the cell types, the non-tumor cell types, and some of the signaling that might be happening there, we do observe that the MCAFs, the fibroblast with the specific phenotypes of the myofibroblast, show significant spatial heterogeneities, and a lot of these heterogeneities were restricted around the tumor stroma local interfaces. As this particular example from the earlier time shows. And also this is interesting because the FGFR is one of the few drugs, or effectively only the drug at this point that is FDA approved for the urothelial carcinoma. So to know that where these activities happen and how these might be helping us stratify the samples is of the therapeutic relevance as well. But then it is not just only the fibroblast. When we look into the macrophages, we do see a lot of these macrophage activities. This is not the first sample. This is, I think, the S9 sample, and there we do see a lot of the heterogeneities, again, in these macrophages in the tumor stroma interface regions as well as in the other regions. And those indicate that there might be a subtype switching happening in the macrophages too, that some of these macrophages in a context-dependent way are switching from the M1 to the M2 subtype. And so that make us think that probably when we look into the tissues, we should not be thinking only about the EMTs in the cancer cells, but there might be these fibroblast-associated signatures, these macrophage-associated signatures, which can make this whole analytics far more complicated. And to throw something more into the mix, we have by accident found a lot of the tumor-associated microbes in the samples. So that was, since I have a few minutes, so that was a very interesting observation. When we tried to do our initial experiments, we found that a lot of the reads do not map to the human, and we thought maybe we need to refine our experiments. We are throwing away a lot of the reads, so there might be a laboratory contaminations. So we went on to map these reads to the non-human species, and that went on to be a whole story in itself, and we found a whole lot of the tumor-associated microbes, and other people have found that too. But to long story short, these microbes tend to influence the cellular metabolism and also immunity. So maybe in this mix, there might be another whole new dimension about how these microbes might be influencing these cell state dynamics in the cancer cells, in the immune cells, and so on. But that's a whole complicated world. So this is my last slide. That so then we'll be thinking about so in which way we think we should be modeling these functional associations in the cell state in the tumor and the non-tumor cells. To what extent having this multi-scale complexity, which is like exponentially rising, what has been necessary and sufficient? And finally, we have historically been trying to target only the cancer cells, but would we be thinking about maybe the adverse events and maybe this whole ecosystem as a whole, and think about these alternative strategies? With that, I stop. Thank
you. Are you using any of these methods for inferring cell-cell signaling directly, cell chat or whatever, all those,
yes,
and plus the spatial transcriptomics extensions of those?
Yes, we are using cell chat.
So that's how you're identifying the signaling.
Yes.
Okay, great.
So you only have 3 transcription factors for the model that are even not include all this EMT regulators. So I see this more like upstream, right? A SMAD3, right? This is a so can we just say, this is really coarse grains model, right?
Yes. So we think that probably there might be many transcription factors that might be involved. And there might be a long tail of how many transcription factors we need to consider. In our experiment, it appeared that with the siRNAs, we could alter the cell state. So we thought that probably for our experiment, this is probably the minimally sufficient, but we think that the actual regulatory network might be much larger. And that's where probably the functional buffering is more important, that maybe once we suppress one transcription factor, others might come up and suppress that activity.
Yeah, that's where you can find the transcription data. I know that you have myofibroblasts,
Yes.
right? That's clearly can have a TGF beta, SMAD3, that pathway or thing, right?
Exactly.
Can you define for us what you mean by functional buffering, mathematically in terms of your landscape?
Yes. So if a gene is making the mRNAs and then making the protein, and ultimately we do care about probably how much copies of that protein is and what functions it is doing. If a protein has got fewer copies, then maybe something else needs to come up more. And that more can come up maybe at an mRNA level for other genes. Yesterday and the day before, we have had some very interesting talks. But also there might be the stability of other proteins might get altered. So I wanted to use an umbrella term that accommodates all those other variations, so that the net functional output of probably that signaling pathway or something has not got altered that much. Sorry for a very fumbling answer.
Or modulation. I just don't know what buffering means in
Or
your.
compensation?
Yes.
When you got into like looking at the tumor stroma interactions, and you saw macrophages and fibroblasts, okay, great, but like then what's happening with the EMP states in the context of that spatial organization?
Right. So there the question is that what is driving what? Is that only the tumor cells are driving the phenotypic alterations in the macrophages in the fibroblast, or it's a reversible and multi-directional switch that is going on there? And so there is a very interesting observations for that is that in the animal models, if you take cancer cells and try to inject it into the mice to make the tumor, in the young mice, it is very hard to make the tumor. In the older mice, it is much easier to make the tumor. There is a framework called adaptive oncogenesis, but if we think about the functional pathways, there might be a lot of the abnormal signaling that would be going on that make maybe the cancer cells and its phenotypic alterations much more receptive in that environment.
Right. I mean, I guess I was asking a much more simpler question, just the associations. Do you see correlations?
Yes, these are only associations. That's why we need much more exact systems to study that and see that what is driving what.
Okay. Yeah, you do. Thank you all very much. Let's continue the questions over coffee later.
Yeah.